Okay, so in this bubble, at this point, is a very specific sequence that allows the initiator proteins to find it. So the initiator proteins have R groups, and those R groups are going to have a very specific tertiary structure. That tertiary structure will match nucleotide shapes. Those initiator proteins are going to come in and bind, and they're going to recruit what enzymes that will open up the DNA and move it along. What do we call those?
Helicase.
Helicase. Excellent. And helicase is unzipping the DNA as it runs down and breaks all those hydrogen bonds that connect it. So do you imagine the initiator proteins are going to bind to a bunch of sequences of G's and C's or a bunch of A's and T's?
A's and T's.
A's and T's. Excellent. All right. So let's imagine this is 5 prime. Okay. So then what is this side?
3.
3 primes. This must be 5. This must be 3. So if we start right there, we're going to have new DNA strands. We're going to make a strand that goes this way, and we're going to make a strand that goes this way. To the one to my left, we're making in what direction? What direction is this right there?
3 to 5.
That's a 3 prime.
5 and 3.
5 to 3, because there's your 5 prime end. This is your 3 prime. So this strand we call what?
A leading strand.
That's the leading strand. Excellent. And therefore, the other strand, we're building towards a 5 prime end. And that's a problem because if we do that, 5, 3, so we're building towards that 5 prime end. So this is going to have to do it in short bits at a time, because I can build right here and go backwards. Okay, so then this is what's called Okazaki fragments. I'm going to stick down a bunch of sequences right there, and we're going to use what to do that? We're going to put down with sequences. What enzyme?
Primase.
Primase. Excellent. Primase is then going to allow DNA polymerase 3 to bind, because you're going to create that 3' end, which then I can go in reverse direction. And we're going to do them in small fragments, because otherwise it would take a really long time. I'd have to unzip the whole strand to run it backwards. And so we're going to do that in short segments. Those are called Okazaki fragments. And eventually what happens is you start creating, I can't figure out how to shut this off so other people can't draw on the whiteboard, because that's what's happening here, and then I erase it. Does anybody know?
My first time seeing the feature, so I don't know.
Yeah, mine too. I tried not to be on Zoom. Anyways, we'll just live with it. And so you end up creating these little fragments here where we have to combine the two sections of DNA. What enzyme combines those?
DNA polymerase?
Is ligase. So we're going to ligase, okay? And then DNA polymerase 1 will come in and replace all of those primer sequences, because those are RNAs. And they're going to come in and replace them with DNA. And so eventually we'll end up with 2 full sets, okay? So we start with 5 prime, 3 prime, 3, 5. So this strand right here runs to our leading. Right here, this strand is also
Leading.
our lagging strands are opposite of that. And so they're going to go like so with those Okazaki fragments. Okay? And those are called lagging. So we have DNA polymerase 3, which is our main DNA. That's going to run in 5 to 3, build your 3 prime. You're going to have helicase, which is unzipping everything. You have ligase, which is ligating the phosphodiester bonds, bringing them back. You're going to have DNA polymerase 1, which is going to replace all of your RNAs. Okay? We feel comfortable with that.
How do you explain backwards again? Because I see them all forward and then backwards. I'm confused with backwards.
So let's look at, this is only 1/2 that bubble, okay? There's your DNA strand. So there's 5 prime, 3 prime, okay? So that means if we're making a new DNA, it can run that direction because that's going to be 3 prime. Well, the problem is if we're trying to go this direction, this guy down here is now going to be 5 prime. So what I'm going to end up doing is putting down fragments, and we're going to build a primer, and we're going to go backwards. Build another primer, go backwards. Build another primer, go backwards. And that's going to allow us to go in the correct direction so we can build on our 3 prime end. Because what side can you build on?
On 3, but if you go backwards, you're still starting on 5, aren't you?
Because we're putting down primers.
Oh.
Because now I have a primer, which will have an open what end?
For 5.
It'll have an open 3 prime.
Oh.
Right? So this is if your primer has a 5 prime end and a 3 prime end. I'm going to now build on the 3 prime end.
And the primer moves with the direction of the fork, right?
Correct.
So
Yeah,
like
it's not-
it builds
it moves-
5 to 3, and then it moves backwards, builds 5 to 3, and then just keeps doing that, right?
Yeah, and just repeatedly doing it. That's why it's called fragments. And it's not that the primer's moving, it's just putting down new primers. So your cell's going to make a ton of primers. So when we did our DNA extraction last quarter and we did our PCR, we stuck primers in that solution. Our master mix was primers and nucleotides. Why did we stick down primers? That's a fabulous exam question. Why did we stick down primers in PCR?
To replace primer sequence with DNA? No.
No.
To initiate the reaction?
Excellent. It needs to know where to start. The primer's going to bind to the correct sequence. It's going to say, hey, we're going to start here. And once it knows where to start, it'll be able to build. But if we don't stick down a primer, will it know where to start building, or can it even start building? No, right?
No.
So only have things start where a primer is at. Okay? All right. So that starts with that. Let's go back and start thinking about transcription and translation. I have the following plays taking place in a cell. Okay? What process am I illustrating right now from our previous questions?
The
Translation.
release factor?
Translation.
Translation.
This is
Translation, okay.
okay. So at the end of my green, that's going to be my N termini. Where's my C?
Well, at the
It's gonna-
beginning of the.
It's gonna be at the start of the arrow?
There's our C termini. So that N is C. And so what's going to come in? So what is this thing right here, first of all?
Ribosome.
Ribosome.
Excellent. That's the ribosome. And what's this?
The promoter.
mRNA.
Not the promoter.
mRNA. Excellent.
Oh, yeah.
Mar, does mRNA have a promoter attached to it?
Let's see. The promoter is in transcription, right?
Correct. And this is trans.
So then no.
Correct. Okay. So this is the ribosome binds to the ribosome binding site. So that could be the ribosome binding site, RBS as well. This is an mRNA. And it's going to bind. And when it binds, it's going to start recruiting things. So if we're already making it, so this would be what right there? What code is right there?
Would it be your ADA or TATA box?
Because that's called what?
Promoter.
That's the promoter. Excellent. So this has to be the start codon.
The initiate.
AUG.
That is AUG. All right. AUG starts translation. Now we're going to have our tRNAs are going to come in and bind there, right? So we have our tRNAs, and that has an amino acid on it. So that's our acetyl tRNAs. That comes in and binds to our C termini so we can continuously build. And this ribosome is going to run down the length of the RNA until it reaches what?
The stop codon.
Excellent. And the stop codon then causes it to fall apart and release, and we can restart translation again, do something new. So let's go back to transcription now. So now I have DNA. There's your DNA. And that's a long chromosome, right? Here is my 3 prime. There's my 5 prime in. So that must be 5. That must be 3. Just like DNA replication, we have to know where to start transcription. So how does a cell know where to start transcription?
When there's a new primer?
No, that's DNA replication.
Oh, yeah.
Promoter sequence.
Promoter sequence, excellent. So if this is our +1, this is our -10, -35. We call that the promoter. That means what direction is the enzyme moving?
Left to right.
Left to right. It's moving left to right. And what enzyme is that?
RNA polymerase.
Excellent. Okay. If this was DNA replication, what binds?
DNA?
DNA polymerase?
Excellent. DNA polymerase. If we were doing DNA replication, it'd just be DNA polymerase. Instead of promoter, it'd be initiator. That's it. Okay, so that's the only differences. So RNA polymerase, though, is only making one strand. So we are not going backwards. We don't have Okazaki fragments. We're not going backwards. So we're only building in this direction because that's our 3' end. So we start at 5, go to 3. And I can start with a 5 because it's RNA. What are our primers made out of? RNA. Excellent. That's how we can build that. Okay, so RNA polymerase is going to move forward, and we're going to get a new strand of nucleotides. So therefore, this side is, what do we call that strand? Excellent. That's our template. We made an mRNA. So what is this side called?
Coding.
Coding.
Coding strand?
All right. This whole thing is called a gene. Okay. How we feel?